Hi, I'm James Kozlowski, and together with Viatcheslav Gurev from IBM Research, Yorktown Heights, New York, we're presenting generative adversarial networks to study the action of a cardiac inotrope on a virtual population of computational models. Now, the objective of our study is shown here, which is to embed mechanistic modeling within this space of deep learning. And in the way that we approach this is to use generative adversarial networks to build populations of mechanistic models that fit certain experimental observations. Those observations in our study involve the length traces of mouse myocytes, which were registered in the experiments with both unloaded contraction in control conditions and under the effect of a positive cardiac inotrope known as omecamtiv mecarbil. Our goal was to find distributions of model parameters that, after push through of the model, fit and reproduce the distribution of features extracted from the experimental traces. Some of those features are shown here, where you can see that the features extracted include the resting sarcomere length, the sarcomere length at maximum contraction, and the time to peak contraction. Here you can see distributions of features extracted from the experimental traces, and these distributions, such as time to peak of contraction and amplitude of contraction, are extracted from the length traces of the myocytes, as I mentioned above. The inferred model parameters are then used to explain these features and to explain the mechanism of the drug action, and transfer parameters between the different experimental settings, again, the undrugged and the drug condition, under different conditions and protocols, such as omecamtiv mecarbil versus no drug. There are many methods to infer model parameters. Many of them are based on MCMC, which is Markov chain Monte Carlo, as well as sequential Monte Carlo. The novelty of our approach is to employ adversarial variational inference by training generative adversarial networks such as those shown here. And then we apply these two types of networks, the first one known as conditional GAN or CGAN, for example, to train the conditional probability of model parameters given experimental observations. This is an example of amortized inference. Another approach known as the regularized GAN or RGAN, in this case, directly incorporates the mechanistic model into the neural network and provides a means to incorporate the prior on parameters, as shown here. So many advantages derive from our choice of using GANs for this adversarial variational inference. The first demonstrated result is of a synthetic problem wherein the model is a nonlinear model of two parameters that takes two parameters and produces outputs which are represented by this heat map. By trying to infer parameters of the model that fit a Gaussian distribution of model outputs, you can see that both CGAN, RGAN, as well as MCMC have to sample from this distribution of model parameters in a very specific way. And what's interesting is that the distance between these distributions of both the model parameters that are sampled versus those used to generate the data, as well as between the outputs of the model and the synthetic data that we're fitting, that those distribution are measured in distance and divergence equally, more or less, for MCMC, RGAN, and CGAN. If anything, CGAN is slightly better. A further elaboration of the GAN, known as transfer GAN, was used in this study. What we did was broke the GAN into 4 discriminators and 3 generators in order to take into account the fact that the model must capture the results from experiments on different myocytes under the omecamtiv mecarbil condition as well as the undrugged condition. And the parameters of the model, furthermore, were known to be affected by the model only in some cases. And in those druggable parameters, a generator could produce both the drugged state of the model, XD, as well as parameters matching the control state of the model for those druggable parameters. Other parameters were shared between the two generators in order to produce a union of them with the drug and control druggable parameters. These, known as shared parameters, were constrained to be sampled to fit both distributions, both with the drug and the undrugged condition. And you could see this is an example of an RGAN as well, where the mechanistic model is embedded into the neural network and is transforming the parameters into model outputs. The results were quite good in that we fit the model to the data. Using the RGAN, we sampled from the model distribution of parameters in order to produce a push-through of the model, which fit the data very well. And as you can see from these traces, the outputs of the model are comparable in terms of the variation that you saw earlier from those traces from the real data. In these 2D distributions, you can see that this also captured the joint distributions between parameters very well, where the samples from the GAN are shown by the dots and the contours represent the actual data. In addition, we were able to transfer parameters into another experiment, such as that shown here, which fits very well data on calcium versus forced calcium relationships in the omecamtiv and the undrugged condition. And so in the end, what we were able to do is use this model of the myofilament and its contraction to fit data from the experimental myocyte, both in the omecamtiv and the control condition, holding certain parameters to be sampled from the same distribution for those two conditions because they were not druggable, and other parameters could be sampled differently for those two experimental conditions to reveal the underlying mechanism of action of the drug omecamtiv mecarbil. This is a novel approach for constructing population of models coherent to the distribution of the observed data. And for the first time, we've incorporated GANs into a variational inference problem such as this. Thank you.